Welcome back, dear students. We have already started with chapter number 2, sexual reproduction in flowering plants, Angiosperm. I just want to remind you that the topic what we have finished, I just list out the name of topics what we have finished. We have finished a flower, a structural or reproductive part of a plant which help in a sexual reproduction. Second, we have discussed the male reproductive part and the female reproductive part, means we have discussed the stamen, carpel, that internal structures have been discussed. We have also discussed a gamete transfer process, what we call it as pollination. We have also discussed the agents which help in a pollination, what we call it as pollination agents. There are two types of pollination agents, biotic and abiotic agents. I hope all of you have referred the notes and prepared it. Now today we go ahead with another topic, and is again important topic which can be asked in examination, and this is called as artificial hybridization. So we start with this artificial hybridization. What do you mean by artificial hybridization and why we are performing this artificial hybridization? You know that, suppose assume that this is a stigma, this is a style, and it is in an ovary. You already know that ovary contains a ovum. It's a schematic representation. Now this is a stamen and this is an anther. From anther the pollen grain is getting released. Okay, and this pollen grain falls onto a stigma. But it is not necessary that the desired pollen grain, means the pollen grain what we want or the product what we want, that desired pollen grain falls onto a stigma. What are the chances that the desired pollen grain falls on the stigma and then results into a fertilization? It is not a certain chances, means we cannot say surely the desired pollen grain only falls on it. So we are also not sure that we get a good variety of a fruit or we get a good variety of a plant. Our next generation might be good, might not be good, depends which pollen grain falls on it. Okay, desired or undesired pollen grain falls on it, depends on the results will come. But now we don't want to be in dilemma. We want to be sure, and to that we are performing a process called as artificial hybridization. In artificial hybridization has very good importance in crop improvement program. Clear? Because here we keep or only a desired pollen grain which is onto a stigma of a carpel, and how we will achieve that we will see. Okay, so artificial hybridization is the hybridization which is performed artificially, not occur naturally. Usually transfer occurs naturally either by biotic agents or the abiotic agent. In artificial hybridizations, the transfer occurs with the interventions of a human being, or we help in the transfer of a pollen grain. Okay, and we transfer only the desired pollen grain, so we get a desired product only. So how it occurs in artificial hybridization? You know that there are two varieties of a plant or flower. One is a bisexual flower and the second one is called as unisexual flower. Let us first discuss how we perform the artificial hybridization in bisexual flower. So assume that this is a bisexual flower. This is a bisexual flower, and that possesses, as it is a bisexual flower, so it possesses both sexual organ, stamen, as well as it also possesses carpel. Both are present. So how we perform the artificial hybridization in bisexual flower? This is a bisexual flower. To perform the artificial hybridization in bisexual flower, we perform the two techniques. The first is called as emasculation. First technique is emasculation. What is an emasculation? Emasculation, that means removing of this anther. This is an anther. Okay, removing of this anther with the help of forceps before it dehisces. Before it dehisces means before the pollen grain releases, we remove the anther from it. So there is no chances that the pollen grain which is released from it can fall onto the stigma. So we protect this stigma. Okay, how we protect the stigma? By preventing a self-pollination. We remove the anther with the help of forceps before it dehisces. Remember the word before it dehisces. Because once it dehisces, means it releases the pollen grain, there is no purpose of protecting the carpet. Already the pollen grain has been released. Okay, so before it dehisces, we remove this anther with the help of forceps. And this process is called as emasculation. Okay, once this anther has been removed, now the second technique is going to be performed. Okay, and this is called as bagging. Which technique is performed? Bagging. What is a bagging? Once the anther is removed, this carpet of a flower has been covered with a bag of butter paper. With the help of bag of butter paper, the carpet or pistil has been covered. So it is protected from the pollen grain of even another flower or the different other flower. Okay, or other species pollen grain that is protected from it. Okay, so that is, this technique is called as bagging. Okay, so what is emasculation? Emasculation, that means removing of anther with the help of forceps before it dehisces. This is called as emasculation. Once you have performed the emasculation, then the carpet has to be covered with a bag of butter paper. This is called as bagging technique. Once it is covered, we allow this carpet to mature. And once it is mature, we remove this bag. Clear? And we keep artificially a desired pollen grain. With that desired pollen grain is obtained from the another plant. Based on that, what characteristic we need in the next generations, we collect that pollen grain and we sprinkle or dust that pollen grains onto a carpet or stigma of a carpet. Okay? Once it is dusted or sprinkled, then it is again covered. So another pollen grain doesn't fall on it. Okay? So this is a technique which is performed in bisexual flower. Okay, and thus we get a desired crop or desired plant or offspring which is produced by this. So in bisexual flower, two techniques are performed for artificial hybridization: emasculation, and the second one is bagging. Now what we are performing in unisexual flower? Now it is a turn of unisexual flower. You know the unisexual flower? Unisexual flower, that means it possesses either stamen or it possesses carpet. Or means either of this organ is present. If it possesses stamen, then we consider it as male flower. If it possesses carpet, then what we call it as female flower. Clear? Now see, so we draw a different flower. Now tell me which flower it is. Just observe it and tell me which flower it is. This is a stamen. So which flower it is? Male flower. And this one is? This is a carpel. Okay, so this is a female flower. Now here male and female flower are the different. Okay, so we don't have to perform both the technique as we have performed in the bisexual flower. In bisexual flower we have performed two techniques, emasculation and bagging. But in unisexual flower, you just have to perform a technique called as bagging. Okay, there is no need to remove this anther because the male flower is a different one, female flower is different one. There is no chances of self pollination. Okay, so here we just cover it with the help of a butter paper. And this covering process, you know it, what it is called? It's called as bagging. What it is called? Bagging. So we cover it. Cover the female flower. We allow the carpel to mature in the female flower. Once the carpel is mature or stigma becomes a receptive, at that time we remove the bag and we sprinkle or dust the pollen grain on it. A desired pollen grain, remember, a desired pollen grain we sprinkle on it. And once we sprinkle, then again we cover it. That what we call it as rebagging. Okay. So here in unisexual flower, we just cover a female flower by bagging. Allow it to mature, allow the stigma to be a receptive, and then we sprinkle the dust of pollen grain, we again rebag it. And this is a procedure which is performed for unisexual flower. So in short, if it is bisexual, two procedures are performed to get an artificial hybridized product. One is emasculation and second one is bagging. And if it is a unisexual, only one procedure is performed, that is a bagging, and again rebagging is performed. Okay, so that's all about artificial hybridization. What is the importance of this artificial hybridization? As in the initiation of topic, I have already told that it has a very good importance in crop improvement program. Here, by desired pollen grain, we can get a desired crop or desired plant only. So that's why it has a very good importance in it. So clear, students? This is a very short note. This is called as artificial hybridization. Okay. Now we move ahead with the next topic, and that is so the next topic is double fertilization. See, we are learning the sexual reproduction, and sexual reproduction, you all know that there are three steps or three basic events in all the sexually reproducing organism: pre-fertilization, fertilization and post-fertilization. We have finished the first process, pre-fertilization, including both gametogenesis, okay, male and female gamete have been produced, and the gamete transfer, how the pollination occurs. Once the gamete transfer, now the turn is fertilization. Okay, the second step. And you know the meaning of fertilization. Fertilization, the broad meaning of it is that's a fusion of male and female gamete, or you can also call it as syngamy in other terms. So how the fertilization occurs and what kind of fertilization occurs in flowering plants or the angiosperm. To understand it, you should know the structure of carpel and detailed structure. Okay, so you can better understand what is called as a double fertilization. So I am drawing a flower. This is a flower. I am just drawing just a female reproductive organ. Okay, because I need just a female reproductive organ. This is a stigma. It's a style, a tube-like structure, what we call it as style. And then finally, the bulge portion is called as ovary. I am drawing a enlarged portion of a ovary or very large ovary because you have to see a lots of things over here. Now, in this ovary, you know that this ovary contains ovarian cavity, what we call it as locules. You have learned the structure of megasporangium or ovule. This ovule or megasporangium contains embryo sac. Okay, so it possesses the embryo sac. Suppose assume that this is only embryo sac, what I have drawn, for easy understanding. Otherwise, it will make a more complicated. But really, what happens? This ovary contains ovarian cavity. Ovarian cavity contains ovules. Ovules like this. These are the ovules that is attached to a placenta by a stalk. Okay, you have also learned the detailed structure of it, but I am just drawing this ovule in an enlarged view over here. Assume that it contains only one ovule. The plant may contain one ovule or many ovule. Like for an example, one ovule is present in mango. Mango has just one seed, one ovule. Even we also have example of watermelon which has many seeds, many ovules. or papaya which has many ovules. But for simplicity or for better understanding, I am just drawing or considering an example which has a one ovule. Okay, the similar thing happens if it has a multiple ovules. Okay, same thing happens into a multiple ovules. Suppose this is a one ovule which is present in ovary. Assume that this is a ovule, enlarged structure. You know that there are two ends. This is called as micropylar end. This is chalazion end. Okay, this chalazion end contains 3 cells, and this is a center. Okay, what we call it as polar nuclei. Now you know that micropylar end, it contains 3 cells. Which are the 3 cells? It contains 2 synergid cell and 1 egg cell. We have already discussed it. 2 synergid cell, and this is very important. 2 synergid cell and 1 egg cell, they combine together form egg apparatus that is present at the micropylar end. 3 cells which are present at the chalazion end, these 3 cells all are of same type, and these 3 cells are called as antipodal cells. And in the center, the 2 cells combines together. So now at maturity, 8 cell becomes a 7 cell, or 8, and that becomes a 7 cell and 8 nucleate. What we call it as, this is a polar cell or polar nuclei. Okay, you already know it. I have shown over here, so I am just rubbing it out. This is a polar nuclei. This is the cell which is only a diploid. I am writing outside. These all antipodal cells are haploid in nature, all 3. This 2 synergid cells and 1 egg cell all are haploid. So these 3, all 3 antipodal cells are haploid. 2 synergid and 1 egg cell, all these 3 are also haploid, but only a polar nuclei, it's a diploid. Polar nuclei, it's a diploid because it is made up of fusion of 2 nuclei. What we have already discussed, okay, during a formation of megasporogenesis or embryo sac formation or female gametophyte. We have discussed it. Now what happens or how fertilization takes place? To fertilize, there is a pollen grain which falls onto a stigma. From pollen grain, a tube will develop a germination. You know that what this tube is called. This is called as pollen tube. Pollen tube contains the cells. Cells are present. Okay, either the cells which are going to be released or pollen grain which is going to be released at a 2 cell stage or 3 cell stage. Whatever, ultimately in the pollen tube there are 3 cells. Which are the 3 cells? 2 male gametes and 1 vegetative cell. Total 3 cells are released into a pollen tube. Now this pollen tube grows towards the ovary. It grows continuously, grows, and then it enters into a ovary through micropylar region. Through which region? Micropylar region. Remember, it enters through a micropylar region. You already know that we have discussed in a pollen crystal interaction the filiform apparatus of it that helps in guiding the entry of pollen tube through a micropylar region. Okay. Now, once it is in a micropylar region, I am just enlarging this much portion over here. This is a ovule. I am just enlarging this portion. Okay, over here so you can better understand. These are the 3 cells. These are 3 cells. What these 3 cells are called? These 3 cells, 2 synergid cell, 1 egg cell, all are haploid. In center, there is a presence of polar nuclei. In these 3 cells which is present at chalazion end, they are antipodal cells. They are also haploid. Now what happens? The pollen tube comes. Entry of pollen tube. This is, assume that this is a pollen tube. I have shown just a half pollen tube. And that contains 3 cells. For easy understanding, I am drawing, actually it is a spherical in shape, but for differentiate or to differentiate, I am drawing a different shape. This is a triangular. Okay, but that's just for a easy understanding. Otherwise, you have to draw it a circular. And you know there are the 3 cells which is present in the pollen tube. Which 3 cells are present in pollen tube? 2 male gamete and 1 vegetative cell. Okay, so what it contains? 2 male gamete and 1 vegetative cell. Once it enters, it breaks this wall. Okay, so micropylar end is through which the pollen tube enters, and these 3 cells are released over here. Okay, these 3 cells are released into an embryo cell. From this 3 cells, 1 male gamete, you know that contains 2 male gamete and 1 vegetative cell. From this 1 male gamete, it fuses with 1 egg cell. This is the first time. You know that fusion, fertilization means fusion of male and female gamete. This is a 1 male, from 2 male gamete, 1 male gamete fuses with an egg cell. Egg cell is a female gamete. So it fuses, and that process what we call it as fertilization. And you know that once the male and female gamete fuse together, what it forms? It forms zygote. So it leads to a formation of zygote by fusion of male and female gamete. Okay, this is a fertilization. Now the another male gamete, because you know that we have a 2 male gamete. The another male gamete that fuses with this polar nuclei. Okay, polar nuclei is a 2n diploid, and another male gametes are haploid. 2n plus n, it makes 3n, and it forms like 2n. I just write over here. 2n, it's a polar nuclear, and this is another male gamete. Both fuse together, 2n plus n, 3n. What we call it as PEN, primary endosperm nucleus. What it called? Primary endosperm nucleus. Clear? 2n is a polar nucleus, n is another male gamete. Both fuse together, form a 3n. Clear? This is, and it forms this 3n cell, triploid cell. What we call it as primary endosperm nucleus. And the first male gamete that fuses with female gamete, the male gamete is a haploid, female gamete is also haploid, and it forms diploid, n plus n, 2n zygote. So here, you know that the fusion occurs, but fusion occurs 2 times. First time, 1 male gamete fuses with another female gamete, forms a zygote. And in second time of fertilization or fusion, another male gamete fuses with a polar nuclei and form a triploid primary endosperm nucleus. So what it forms? Ultimately, 2 products are formed. PEN, primary endosperm nucleus, which is triploid in nature, and the second which is going to be formed is a zygote, which is going to be formed, which is diploid in nature. So at the end, 2 products are formed in a angiosperm or flowering plant, zygote and primary endosperm nucleus. Okay, zygote is formed nearer to a micropylar region. Primary endosperm nucleus is present nearer to a center of a embryo sac. Okay, so here the fertilization fusion occurs 2 times. So that's why this is called as double fertilization. What it is called? Double fertilization. Clear? So that is a double fertilization. I again repeat how the double fertilization, why it is called as a double fertilization. Double fertilization is observed in angiosperm or flowering plants. In one case, the pollen tube contains 2 male gametes and 1 vegetative cell. These male gametes and vegetative cells, they are released into a embryo cell. From these 2 male gametes, 1 male gamete fuses with an egg cell, female gamete, egg cell, and that forms a zygote. Male gamete is a haploid, female gamete is also haploid. Both together form a diploid zygote. That is the first product which is going to be formed in a first fertilization. So now 1 male gamete is utilized. 1 is left. That another male gamete which is left, that fuses with polar nuclei 2n. Clear? 1 male gamete fuses with a polar nuclei 2n, and what it forms? 3n, primary endosperm nucleus. And this is that contains here the 2-time fertilization. So that's why this process is called as double fertilization. Okay, so now at the end of fertilization we have 2 products. One is zygote and the second one is primary endosperm nucleus. Okay, next We are not starting the next topic, but I just briefly inform you so you can come to know that, or you will remember that what will happen in the next session. So at the end of double fertilization, what we have? We have zygote. This zygote is diploid. This is a product of first fertilization. And in the second fertilization, we have a product called as PEN, primary endosperm nucleus. And that is 3N, triploid. Okay, primary endosperm nucleus. This is formed by a fusion of male and female gamete, both together fused together form a zygote in a fertilization. PEN is formed by a fusion of polar nuclei 2N and the another male gamete, both fused together form a PEN, primary endosperm nucleus. As there are two-time fertilization, first time and second time, two-time fusion occurs, so that's why it's what we call it as double fertilization. But ultimately we have two products, one zygote and the second one is PEN. Now from this zygote, what is developed? Embryo will develop. Okay, and usually it's a 2N. And from this PEN, what it is developed? What we call it as? PEN, what we develop? The endosperm is going to be developed. Okay. So from zygote, what develops? Embryo. From PEN, primary endosperm nucleus, what is formed? Endosperm is going to form. Okay. So in the next session, we will discuss how PEN converts to an endosperm. Okay, and that includes under post-fertilization events. And how zygote converts to an embryo. That's post-fertilization events. Now fertilization over, after fertilization the process is occurs what we call it as post-fertilization processes. So after fertilized over, the zygote converts to an embryo, PEN converts to an endosperm. Okay, but first we will discuss this one, PEN converts to an endosperm, and then we will see how the zygote converts to embryo, because the process occurs in the sequential manner in an endosperm for a certain reason. Okay, I will tell you the reason in the next session, but till then, first we will learn that PEN to endosperm, and then we will learn from zygote to embryo formation. Both process we will discuss next time. Okay, so keep learning, keep watching, like and subscribe the video, and thank you for watching.